Good afternoon and welcome to the NIH's Wednesday afternoon lecture series. It's my absolute honor to be here for the first time, and I'm looking forward to the lecture. Today's lecture honors the legacy of Margaret Pittman, the first female laboratory chief at the NIH. Established in 1994, the lecture is given by a researcher who exemplifies the intelligence, scientific excellence, and drive that made Margaret Pittman a leader. It's my pleasure to introduce our speaker for today's lecture, Dr. Carolyn Bertozzi, who's going to come out in just a minute. I should say a former colleague of mine from Stanford. Dr. Bertozzi is the investigator at the Howard Hughes Medical Institute. She's the Baker Family Director of Sarafan ChEM-H, and Ann T. and Robert M. Bass Professor of Chemistry, and Professor by courtesy of Chemical and Systems Biology and Radiology at Stanford. Before I tell you her background, I should let you know that if you are attending this lecture for CME credits, the code is, and it's on the screen, will be 57692. And if you're watching this online and would like to submit questions, please click on the Send Live Feedback button, and your question will be relayed to the speaker at the end of the lecture. You can submit questions anytime during your lecture. We are absolutely honored to have Dr. Bertozzi here today. She is a chemical biologist and Nobel laureate known for her research interests that span the disciplines of chemistry and biology. Her work primarily focuses on cell surface sugars important to human health and disease and how to use biocompatible chemistry to modify those sugars. She coined the term bioorthogonal chemistry for click chemical reactions compatible with living systems. Her research targets changes in cell surface glycosylation associated with cancer, inflammation, and bacterial infection, and uses this information to develop new diagnostic and therapeutic approaches, most recently in the area of immuno-oncology. For her groundbreaking work, she received the 2022 Nobel Prize in Chemistry, sharing it with Morton Meldal and Carl Barry Sharpless. Dr. Bertozzi was awarded the MacArthur Genius Award at age 33 and was the first woman to receive the Lemelson MIT Prize. She's a member of the National Academy of Sciences, the National Academy of Medicine, the National Academy of Inventors, and among many other honors and positions. Her research is reflected in about 440 publications across a spectrum of chemistry and biology journals, and she has mentored 81 former Ph.D. students and 58 former postdoctoral fellows. She completed her undergraduate degree in chemistry from Harvard in 1988 and her Ph.D. in chemistry from UC Berkeley in 1993. After completing her postdoctoral work at UCSF in the field of cellular immunology, she joined the UC Berkeley faculty in 1996. In June 2015, she joined the faculty at Stanford University as an Institute Scholar in Sarafan ChEM-H, which stands for Chemical Engineering and Medicine Applied to Health. Please join me in giving a warm welcome to Dr. Bertozzi.
Hello. Hello. Thank you so much for coming today. Thank you, Dr. Bhattacharya, for the very kind invitation and the introduction and for hosting me today. I've already learned so much just from my first morning here, interacting with the scientists and kind of finding out what's at the leading edge of science, which often comes right out of the NIH. You guys are an amazing group of physicians and scientists and staff. You do so much to serve your country. I'm eternally grateful for all the support that I've had from the National Institutes of Health. I'm going to talk about some work that was funded by the NIH, starting when I was a brand new assistant professor. At the time, that was at UC Berkeley. So this kind of takes me back to the late 1990s. But hopefully, by starting in the early days, you'll get to understand the progression of a program that started very much as a basic science project, but eventually evolved in a more translational direction to the point where there are now human therapeutics in clinical trials that leverage some of the chemistries that I'll talk about today. Before I get into any of the details, I would also like to sort of share the honor of helping you celebrate the life and the career and the impact of Dr. Pittman. I read a bit about her last night. She clearly was an incredibly influential figure in the field of microbiology and vaccine development, clearly had a huge impact in her academic positions as well as here at the NIH. So it's an extra honor for me to be on this stage recognizing her contributions. So with that, let's talk a little bit about chemistry. And I always like to get a show of hands so that I understand my audience. How many people here would consider themselves to be a chemist? All right. How many people here would consider themselves to be a not chemist? It's actually quite a few. Okay. There's nothing wrong with that. Nothing to be ashamed of whatsoever. That's a joke for chemists. All right. So regardless of whether you're a chemist or biologist or some other type of scientist, you'll probably recognize this gadget. When you take organic chemistry lab courses, and I think the laser pointer is supposed to work, but seems to be silent. Oh, there it is. Ah, there it is. When you take an organic chemistry class, which I teach at Stanford, and you're in the laboratory and you're doing a chemical reaction, you'll be using a gadget like this one. So this is a round bottom flask, highly engineered glassware, and the logic of this glassware is it allows you to control every parameter of the reaction. So you, as the chemist or the chemistry student, you control exactly what goes into this flask, the concentrations of those reagents, the solvent that you dissolved the reagents in, which matters a lot when you're doing organic chemistry. You even control the atmosphere above the liquid. Sometimes you'll purge this atmosphere to get rid of the oxygen and the moisture from the atmosphere. You even control the rate at which the stirring occurs. You control the temperature with this little bath down here. And all of this very rigid control allows you to form all kinds of bonds using all kinds of reagents. And we have giant telephone books full of lists of organic reactions that you can do if you're doing them in this kind of setting. Back in the 1990s, in the early 90s, as a postdoctoral fellow in an immunology lab, learning about all of these mysteries of biology that were difficult to answer at the time and to understand, I started to become interested in the idea of doing chemistry in much less controlled environments, and quite specifically, to do chemistry in the environment of living systems. At the time, I was thinking about doing chemistry on living cells in a dish, or in the body of model organisms in the laboratory. But you don't have to extrapolate too far to start thinking about what if you could do chemical reactions to form new bonds, new forms of matter inside the human body. And if you could do that, what opportunities might open up in the world of therapeutics development or diagnostics technologies? So it was that kind of thinking that ultimately led to the development of this new, at the time, brand of chemistry that we called bioorthogonal chemistry, which is a bit of a mouthful. And people often ask me, what does that mean? And so here's a definition that is not in the Webster's dictionary, but maybe someday it will be. Literally, bioorthogonal chemistry means chemistry between reagents that neither interact with nor interfere with a biological setting. So they're literally orthogonal to biology. And I recruited a whole brand new group of students when I started my first faculty appointment at Berkeley, and we started taking inventory of what we called biological reactivity space. So the notion is that there's a whole universe of chemistry, all the different functionalities you could imagine from the elements on the periodic table. But only a small portion of those chemistries occur in biology. As vast as that biological chemical space is, and you could just take inventory of your body. All the chemicals in your body are in this galaxy of biological reactivity space. But somewhere else in the universe, we thought there could be a different galaxy, a galaxy of chemicals that don't exist on the planet Earth, that are not in the human body. Maybe they're chemicals invented by chemists that nature hasn't even arisen yet. And if we could identify reactions amongst these bioorthogonal chemicals, those are the kinds of chemistries we might be able to do without interference in living systems on Earth, in the human body in particular. So that was the notion. Now, as abstract as it sounds, I had a very specific application in mind at the time. I was at UCSF in the medical center, working in an immunology lab, and my goal was to study changes in the pattern of cell surface glycosylation. specifically at sites of inflammation. So this is my opportunity to just kind of give you a little textbook drawing of how people depict the surface of cells and the patterns of glycans on the cell surface. Most of you will know cells are bounded by a plasma membrane. Anchored to that membrane are molecules like glycolipids and glycoproteins. In fact, most of the proteins on the plasma membrane are glycoproteins. They have sugars attached to them. And glycobiology is an area that I developed an interest in really starting as a grad student, but really blossomed in my postdoc years. And it turns out that the structures of these glycans, which, by the way, the glycoscientist uses a symbolic lexicon to describe these structures. They're made of simple sugar building blocks, and the building blocks are in this little table. Each building block has a shape and a color that's discrete. And as you can see from the cartoons, these shapes and colors get linked together in different patterns. And the totality of all of these glycans on the surface of the cell, we call that the glycocalyx. Now, even back in the 80s and 90s, when I was in my training years, it was clear that the glycocalyx changes its composition when diseases arise, almost inevitably. And this had been studied most prominently in the setting of cancer. So even back then, it was clear that there were these shifts in the pattern of the cell surface glycocalyx, as we call it, in the setting of cancer. But it was really hard to characterize these changes. And no one really had any clue about the functional significance. So this was very early days in the field of cancer glycoscience. And I had this idea that it would be really cool if you could actually image sugars on the surface of cells, because if they're changing and you can image them, maybe you could detect a tumor in the human body by magnetic resonance imaging or PET or CT imaging, if you had a way to light up these sugars, which would differentiate the healthy cells from the cancerous cells. And there was one particular phenotype, a glycophenotype, as we would say, that's very widespread amongst cancers. And it's the overproduction of glycans that terminate with a simple monosaccharide unit called sialic acid. So going back, if I can do that, back to the vernacular. So you look down here, here's the chart. This is the cheat sheet for the sugars and their little symbols. The sialic acid is this pink diamond. And healthy cells will have a certain pattern of sialoglycans. Let's call it the healthy pattern. Sometimes I use landscaping metaphors to talk about these things. So I would say that's a well-manicured garden of sialic acids. But the observation is that in the setting of cancer, that well-manicured garden just overgrows, and it becomes a tropical jungle of sialic acids. We would call that a hypersialylated glycocalyx. Now, if this is so common in tumors, you might think, well, if we could image sialic acid, if we could just find a way to light up that sugar in some kind of clinical or diagnostic imaging platform, that might be a way to differentiate the tumor tissue from the healthy tissue, which is a very powerful capability if you're trying to treat cancers or monitor cancers or diagnose cancers. But, back in the early 1990s, we did not have any way of imaging sugars. They were one of the biomolecules for which there was just no tools to light them up in living systems. So, as it happens, oh, by the way, sorry, I have a little shameless plug. I'm giving a talk tomorrow also at NCATS in Rockville. And in that talk, I'm going to tell you why this happens. So don't ask me that now if you plan to go to that talk tomorrow, because you'll spoil it. In this talk, I'm just going to talk about trying to image this difference without why, okay? But come tomorrow if you want to know why. All right. So I went to a conference, and this is why conferences are so great. I was still a postdoc, kind of working on my job applications. I landed in a conference in Southampton, England. It was a kind of a boutique meeting, off the beaten track, didn't really know anybody else there. But that's where I met Professor Werner Reuter, who was a German biochemist who had dedicated his life to studying the biosynthesis of sialic acids. And he gave a talk, and in that talk, he presented some unpublished work in which his group had found that the biosynthetic pathway for sialic acid will tolerate subtle chemical modifications to the metabolic precursors. And quite specifically, what he found is this sugar, which is known as N-acetylminosamine when the R group is a methyl group, that's a normal precursor for sialic acid. There are 6 enzymes or so that transform the minosamine into the sialic acid, and a couple more enzymes get that sialic acid attached to those glycans on the cell surface. And what his lab has done was replace the acetyl group at this position with 1-carbon homologs. And by that I mean propanoil, butanoil, pentanoil. Just stretch the carbon chain a little bit at a time. And as long as those modifications were not too extravagant, all the enzymes in this pathway would say, fine, free pass. So what that means is you feed a cell this modified precursor sugar, literally just dump it into the cell culture media, and wait a couple hours, and the corresponding altered sialic acid with that same side chain appears on the cell surface. And when I saw that talk, an idea arose. What if you could embed within this sugar some kind of a chemical handle, something inert, something that would not interfere or interact with the cell, let that chemical handle make its way to the cell surface sialic acid, and then do chemistry with an imaging probe. So I came back from that conference and sat down and wrote my job applications. And this is literally a graphic from one of my job applications. You know, you write a proposal when you're applying for these faculty jobs. And by the way, this graphic was very state-of-the-art, circa 1994, not so much now. But in that proposal, I wrote, okay, what we're going to do is develop a glycan imaging technology where the first step would be to feed cells a chemically altered sugar that makes its way through biosynthesis to the cell surface glycan. And when that's done, we will then add a probe molecule. And the idea was that there'd be a chemical group, let's just call it X for now, on the sugar, and a complementary chemical group, let's call it Y, on the probe. And X and Y would find each other within this living system. This would have to be on live cells, maybe live organisms. And a chemical reaction would occur that basically attaches the probe to the sugar. And now you can visualize the sugar. That was the idea. So I got the job at Berkeley, and I realized immediately that we would have to invent a new type of chemistry to do this. Because for chemistry to occur in a living system requires this concept of bioorthogonality, but no such chemistry existed at that time. So really the first order of business was to try to invent new chemical reactions that would be useful for an experiment like this one. So there I was. This is UC Berkeley, and this is me in, I think, 1996, so I was about 30 years old at the time. If you don't recognize me, that's why. Almost 30 years have passed since that time. And I applied for my first grant from the NIH. And after a couple of false starts, a couple of revisions, my grant was funded through the National Institute of General Medical Sciences. And that's when I launched this search for bioorthogonal chemical reactions. So thank you to the NIH. Okay, so what did we do? I won't go through the whole odyssey. I'll just cut to the chase. We invented a bioorthogonal chemistry. We published that back in the year 2000. And this was a chemistry that we called the Staudinger ligation. That was in reference to a famous German chemist from the early 1900s who had first published on the reactivity of this chemical group called the azide, where there's 3 nitrogens in a row, with this chemical group, which is a triaryl phosphine reagent. And there is a chemistry by which a person can reduce azides to amines using triphenylphosphines. That's the Staudinger reduction. How many people here have done a Staudinger reduction? I knew it. A couple people. All right. It's an oldie but goodie. And I had done this chemistry myself when I was a grad student for various reasons, but it occurred to me that a slight modification of the classic Staudinger reduction might convert this reaction into a ligation chemistry, where 2 things can be joined together. And that's exactly what this is. And this is a chemistry that you can do on cells, no problem. There's no harm to the cell, no interference with any of the biological chemicals. Azides and phosphines, as far as we know, were made by humans. They're not in our bodies. They're not natural chemical groups. And this chemistry was so well tolerated in biological settings that we could even perform this in the body of a mouse. And we published, this was the work of two former students, Jen Prescher, who's now a professor at UC Irvine, Daniel Dubey, who's now a professor at Bowdoin College. And what they showed is you could inject a precursor of sialic acid with an azide on that side chain. And this sugar would get metabolized in the body of the mouse. And a couple days later, you would find the azidosialic acid populating the cell surface glycans. And then you introduce your phosphine probe. Here's an example where the phosphine is here. That will react with the azide. And then the rest of this is just a probe. It's a flag tag, something that you can find and quantitate on the cells after the reaction is complete. So we spent many years playing with this chemistry, doing interesting things with it, publishing with it, and so on. But we realized pretty quickly that this was not the ideal chemistry for imaging applications, for the simple reason that it's a rather sluggish reaction. It goes a little bit too slow. It's fine for reactions to go slowly in the round bottom flask because you can heat them up and you can let them go for days and days and days. In a mouse, you can't heat it up. Well, you shouldn't anyways. And furthermore, you can't just wait for days and days and days because the mouse will clear these reagents out of its body before the chemistry has had a chance to occur if the reaction is too slow. So in the early 2000s, we kind of went back to the drawing board and started thinking about other chemistries we could perform, ideally preserving the azide as one of the reactive partners, just because we loved it so much. It's so small. It has such great stability in biological systems. Lots of human medicines have azides in them, so we know a lot about the metabolism. They're very inert in the human body. And it turns out that azides have reactivity with other kinds of groups besides these phosphines, one of them being alkynes. Now, alkynes were well known, also some foundational chemistry from Germany from the 1950s and 60s, well known to interact with alkynes to do what's called a cycloaddition chemistry to form a triazole. That's what this 5-membered ring with the 3 nitrogens is. This is a chemistry that also was well known to the chemical community, but not ideal for bioorthogonal applications because it's even slower than the Staudinger chemistry on its face. But in the year 2002, these two synthetic organic chemists, Barry Sharpless, who's at Scripps in the US, Morten Meldal, who's at University of Copenhagen in Denmark, independently and simultaneously discovered that you can speed up this cycloaddition dramatically by adding a copper catalyst. And this copper-catalyzed azide-alkyne cycloaddition became popularly known as click chemistry. And one thing I've learned over the years is that for the biologists in the audience, you've probably not heard of any chemical reaction by name other than click chemistry, right? So how many biologists have heard of click chemistry? It's like, get your hands up, everybody. Okay. And how many biologists have done click chemistry? Yeah. This is one of the few chemical reactions that even a biologist can do in the laboratory. But that says something. That tells you that this is robust, foolproof. You can spit on that reaction and no negative consequence. Sorry to the competitive pre-meds of the world. That's an inside chemistry joke. And when this paper came out, we got really excited because this is such a fast reaction. It doesn't take days. It takes minutes, maybe an hour at most. The problem, though, was this copper catalyst. That copper catalyst is toxic to cells. So while click chemistry is useful to join things together in very complex, even biological settings, it's not useful if the objective is to study something that is alive, whether it's cells or organisms, okay? So that wasn't going to solve the problem for us. But it turns out there are other ways of accelerating the rate of an azide-alkyne cycloaddition that don't require a heavy metal catalyst. And basically the strategy we took was to take that alkyne, that triple bond, and basically confine it in an 8-membered ring. And when you do that, you induce ring strain. And the chemists here will know when things are strained, they're uncomfortable. Molecules want to release that strain. And when this 8-membered ring, cyclooctine, reacts with the azide to form the triazole, a lot of that strain gets released. And that drives the kinetics of the reaction. So this copper-free click chemistry, as we call it affectionately, is something that you can do in living systems without having to worry about toxicities. And by the way, Barry and Morton and myself, as Dr. Bhattacharya mentioned, were recognized for this suite of chemical reactions with the Nobel Prize in 2022. Okay, so now we had figured out the kinetics problem. We had a new bioorthogonal chemistry, and the objective, once again, was to get those azides into biological molecules, cells, organisms, in the sugars, and then introduce cyclooctine probes with fluorescent dyes, or imaging probes more broadly, let these two chemicals click together, and then image the products, okay? And putting the azides into the sugars turned out to be the easiest part, because we had this great precedent from Professor Werner Reuter's earlier work. So, like I said, we had the azide on our minosamine precursor. We fed that to cells. The cells did the hard work and built an azidosialic acid, which ends up on the surface. But you can do this with pretty much any sugar. So, for example, we can put azides into fucose. Any of the glyco people here will recognize why we'd want to do that. That's another interesting sugar biologically. And also into N-acetyl-galactosamine, or GalNAc, by just putting the azide onto the acetyl group. And folks here who study the mucin family of glycoproteins might already have availed themselves of this technology. Who here has fed cells GalNAs? Yeah, all right, Dr. Hanover. So we kind of figured it out. All right, now we got to do some real imaging experiments. And the organism we started with, it's a popular choice for optical microscopy, was the zebrafish. Zebrafish are translucent, so that's helpful. You can see right into their bodies. Also, they're vertebrates, and their developmental program is very similar to all other vertebrates, including humans. And as people here are certainly aware, the programs of embryogenesis very much mirror the programs of cancer development and dissemination. Many of the same genes that we associate with malignancies are transiently expressed during embryonic development. That's also true for cell surface glycans. So this was a good place for us to start. And for those of you who have not spent time looking at zebrafish embryos, it all starts with a single cell that you fertilize in a dish, in vitro fertilization. And the embryo starts to develop from that moment on. And at the end of 5 days, you have a young adult fish. And you can just watch the developmental program unfold in real time during this 5-day period. So we started doing experiments of this type, where we would take these embryos, we would bathe them in media that had sugars with azides in them. We would let those sugars get taken up, metabolized, integrated into the cell surface glycans. And then at various time points, we would add a dye, different colored fluorophores linked to a cyclooctine. This one happens to be a difluorinated cyclooctine. And the click chemistry would occur. And then you could do it again and again and again to your heart's desire, right? So we could take these fish, do a little washing step, and then bathe them in a new batch of azidosugar, which would introduce a new batch of azides, and then you could click on a different color fluorophore. And through this kind of experimentation, we could look at different sugars, different time points throughout the course of development. So images like these became sort of like the dominating data sets in our lab around those years. This now takes me up to about 2004 through 2008. Up here is the head of a 5-day-old zebrafish that was labeled with 3 different sugars at 3 different time points. You can see the colors are distributed in interesting different ways. You can distinguish the pattern of glycosylation in the upper respiratory tract, right, in the nose, which is distinct from the pattern in the epidermis, for example, or in the cartilaginous tissue. You can back up to very early stages of embryogenesis. This ball of cells is a 24-hour-old embryo. And a lot of cell division is happening in these early stages. Cells are making their decisions about what tissue fate they will adopt. And we captured some interesting phenomena. So, for example, every time we found a cell going through a round of mitosis, we discovered that the cell surface sugars were redistributing themselves. And below is a blow-up of one of these events of mitosis. So let's see if I can get this to play. Here we go. Okay, so this is a blow-up of a mother cell who has duplicated her chromosomes and has now divided to 2 daughter cells. It's the same frame in each of these panels, but we're looking at different molecules with different colored fluorescent dyes. So on the left, these are cells in which the fluorescent dye is on the DNA. That's why you're looking at the chromosomes. And looping back to the beginning, here they are. They duplicated, classic microtubule segregation. That's what cell division looks like through the lens of DNA. Jump over a panel. In this frame, we have a membrane protein labeled with a fluorescent protein, a genetic fusion. And you can see that the membrane starts as basically circling the mother cell, and as it divides, you get new membrane forming between and ultimately two daughter cells. That is what cell division looks like through the lens of the membrane. Okay, now, for the first time, we got to see what cell division looks like through the lens of the cell surface glycans. And it was kind of surprising. You can see they look like kind of like a fuzzy halo. You can picture all those trees and the grass and that landscaping on the cell surface. But as the cell gets to the very late stages of cell division, these glycans are no longer uniformly distributed. They start to pile up right here at the junction between the two daughter cells. And it turns out they are contributing to segregation at this stage of cell division. And we spun out a whole project sort of studying this fundamental contribution of these glycans to late stages of cell division. So, it's kind of like you turn on a new spotlight, you shine it in a new place, and you see a new thing. And that was our experience using these bioorthogonal chemistry tools. And by the way, the last panel is just an overlay of the three first panels where they're false colored. Okay. So, suffice it to say that at this point, we had developed a technology, a strategy for imaging sugars on the surface of cells. We had developed some tools that we could use in animal models, and we spun out a lot of these technologies into companies. Some of them were licensed by companies interested in clinical diagnostics. A few companies I co-founded with some students from my lab. But I thought it would be fun to kind of fast forward and tell you a very recent story, also basic science discovery, that was enabled by bioorthogonal chemistry. And it takes me back to the glycocalyx, or the cell surface. Every textbook you read will tell you that there's two kinds of glycomolecules on the surface of a cell. There's glycolipids and there's glycoproteins. Okay? That has been the standard state of the art of knowledge for more than 100 years now. Okay? Every textbook will tell you this. It turns out that's not all. A couple of years ago, we discovered that there's another biomolecule that also gets glycosylated and also presented on the cell surface, and it's RNA. All right? So check this out. Ryan Flynn was a former postdoc in my lab. He came from a Ph.D. training in Howard Chang's lab at Stanford, who is a very famous RNA biochemist, who's now the head of research at Amgen, just recently left Stanford for Amgen. So Ryan came to my lab with deep expertise in RNA biochemistry. And he asked me one day, he said, how come all the other molecules get to be glycosylated but not RNA? How could that be true? And I said, this is the way it is. That's how life evolved. And he says, all right, has anyone actually looked? And I was like, I don't know. And we certainly hadn't looked. And as far as I could tell, no one in the world had thought to look. And people in the audience who are the cell biologists among us are probably thinking, why would you look? Right? Doesn't make any sense. We all know RNA is in the cytosol nucleus. These glycosylation events are taking place in the lumen of the ER and the Golgi compartment. They're physically distinct, topologically separated. Those two worlds don't touch each other. Why would you ever think that RNA could be glycosylated? But fortunately, Ryan thought, okay, if no one's looked, let me be the one. And then we can answer the question once and for all. So he used azido sugars as probes. And since he was really good at purifying RNA and characterizing it, he thought, if I get a sugar probe into my RNA, maybe my RNA is glycosylated. So he did that. He took some cells. He fed them azido sugars, a sialic acid, actually. He then did some RNA abstraction and purification steps, and then clicked onto that purified sample a biotin probe using the copper click chemistry. And then he analyzed the products of that click chemistry using various gels and blotting techniques. And he was quite surprised to see there is, in fact, a species there that is RNA carrying the sugar. He ran a sucrose gradient to separate out large RNAs from small RNAs. And what he found is that all of this sugar-labeled RNA It's actually mapping with the small RNAs, but it runs on a gel like a large RNA. It has an anomalous migration on a gel. Could it be glycosylated RNA? So for the next 2 years, we basically spent every day thinking about how to shoot down this hypothesis because it just seemed impossible. But at the end of the day, we proved to ourselves and ultimately to the community that there are glycoRNAs on the surface of our cells. In this very first set of experiments, the type of glycoRNAs we identified, they were all small structured RNAs, like yRNAs, snow RNAs, and quite a few tRNAs. And the glycans that we first identified were highly branched, also highly sialylated, N-linked glycans. And we published this back in 2021. I think the community was a little skeptical at the time. By the way, it took 4 rounds of review and 7 reviewers to get this thing published in Cell. One of the things we report in that paper is this intriguing observation that glycoRNAs on the cell surface can engage certain members of the Siglec family of immune checkpoint receptors. So it's early days, but we speculate that this engagement might be part of an immune modulatory or immune training event. And perhaps these cell surface glycoRNAs play some role in immune regulation. That's kind of an open question. Most importantly is we put a lot of work into identifying the specific chemical linkage that connects the glyco and the RNA. And one of the linkages that we identified was basically glycosylation of a well-known base modification that's highly conserved in small structured RNAs, including tRNAs. And that's the so-called ACP3U modified base, which has this big chemical name. But in a nutshell, we have these ACP3U residues highly conserved in specific parts of your tRNA. It varies from species to species, but in mammals, there's always an ACP3U right here in this particular loop. And by the way, here's the chemical structure. You can see that it's basically uridine with this extra amino acid bit hanging off of it. And there's a biosynthetic pathway that's been characterized by which it occurs on the base when it's already in the tRNA. So you get the modification. So with a collaborator named Ben Garcia, who's like a phenomenal world-class mass spectrometrist, Ryan's lab, my lab, and Ben's lab were able to put together the detailed chemical structure that links this N-linked glycan to the tRNA. And it looks like this. Here's the base. Here's the modified base. There's an amino group here. And then from then on, it looks just like a glycan that you would find on your favorite neighborhood asparagine residue on a protein. Shocking, okay? And Ryan has now done a lot more work on this. He's now a young professor at Harvard University, and he is basically building out the story of glycoRNA biosynthesis, trafficking. How do these RNAs get into the lumen of the ER? That's where they're getting glycosylated. Is this form of N-linked glycosylation a primordial form that was later mimicked by proteins with their asparagine residues? Or the other way around? I mean, ACP3U is highly conserved in all domains of life, right? This might be something that's very ancient. So his lab is really picking this apart, and I would encourage you, if you just do a little PubMed search of glycoRNA, you'll get the world's totality of papers. There's about 40 as of last night. Ryan has a pipeline that's coming out over the next few months that's going to blow your mind. And the immune system is definitely a big part of this. Okay. But anyways, hopefully you'll see that this is the kind of discovery, basic science discovery, that can come when you have a tool that allows you to open a window to a new type of molecule. Okay, so let's revise the textbooks. Hopefully, in the next generation of textbooks, you won't just see the glycoproteins and the glycolipids, you'll actually see glycoRNAs attached to the surface of cells, and as a preview, anchored by cell surface RNA-binding proteins. All right. So in the last part, I'm going to give you a couple of vignettes that illustrate how bioorthogonal and click chemistries have started translating into the world of human therapeutics. And already, you can Google around on websites, and you'll find a number of companies will advertise themselves as using bioorthogonal chemistry as a platform. A very popular application is in making kind of next-generation antibody-drug conjugates. This is an area that my lab has also worked in. There's a company that's using these chemistries to make vaccines. They're glycoconjugate vaccines for pneumococcus, for example. There's cell therapy companies. I'm going to tell you a little story there. But people who make any kind of complex conjugate probably are availing themselves of these click chemistries and bioorthogonal chemistries. So ADCs, a quick vignette. These are amazing molecules. They're conjugates between a tumor-targeting antibody and a toxic payload connected by a chemical linker. This is an area that I've been following really since it caught fire back in the 1990s and FDA approvals in the 2000s. Right now, there's at least a dozen antibody-drug conjugates that have been approved by the FDA, and the clinical trials, especially in China, are in the thousands right now for next-generation ADCs. People have spent the last 30 years systematically solving the problems. We've figured out how to make better antibodies. We have figured out what kind of payloads work best in ADCs. We've designed really clever linkers that are stable in serum but fall apart inside cancer cells. But the area that hadn't really been disrupted yet, to my mind, was how you actually do the chemistry between the protein, which comes from recombinant production, and the linker drug, which comes from chemical synthesis. And right now, all the ADCs that have been FDA approved use what I would consider to be old school bioconjugation. That means they're doing the chemistry on lysine side chains or cysteine side chains without much control over the sites and stoichiometries of the modification. So this is the kind of problem that bioorthogonal chemistry is really well positioned to solve. Because if you have an antibody, which might typically have 70 or 80 surface-exposed lysines, there's no way for you to pick just the 2 or 3 or 4 that you want to attach a drug to. And you certainly can't attach drugs to all 70. You'd kill the molecule. But if there was a way to introduce a new chemical group, a bioorthogonal group, that neither interacts nor interferes with any other side chain functionality, then you can direct the chemistry exactly where you want it. You can put a payload, here, or here, or here, or here, as long as you have a way to introduce this chemical group at the sites and with the ratio of interest. So we put some thought into how to leverage bioorthogonal chemistry this way, and ultimately developed a technique that we call the aldehyde tag method for making site-specific antibody-drug conjugates. And it starts with the use of an enzyme called the formylglycine generating enzyme, which will oxidize the cysteine side chain in a short amino acid sequence that's only 5 residues, and convert it to formylglycine, which basically has an aldehyde. So in other words, if you clone this little sequence, which is cysteine X proline X arginine, you clone that into your protein of interest at your site of interest, this enzyme will introduce an aldehyde there. Now that there's an aldehyde, you can actually do chemistry on that aldehyde that doesn't interfere with the rest of the protein. So on the ground, what we would do with an antibody is clone that motif into the gene sequence of either the heavy chain or the light chain, depending on where you want it, express those plasmids in CHO cells with a plasmid that boosts this enzyme's expression. And the CHO cells will just secrete the antibody with aldehydes right there, ready to go at the sites you engineered. We invented a chemistry, also based on some ancient German chemistry that I won't go into, but it's a ligation chemistry that allows you to attach a linker of interest and a payload directly to those sites. And for the chemists here, always somebody asks me, what's the mechanism? Here it is. Basically, this dimethylhydrazinoindole reacts with the aldehyde to form an iminium ion, and then you get an intramolecular electrophilic aromatic substitution. The people in the audience who are hardcore organic chemists will recognize this as an adaptation of the Pictet-Spengler ligation or reaction. And ultimately, that forms a carbon-carbon linked product. It's rock stable, will not fall apart even in the human body. So we formed a company called Redwood Bioscience by myself and also my former student, David Rabuka. And eventually that company was acquired by Catalent. And so now this is one of the offerings of Catalent Biologics. They partner with companies who want to make ADCs. You just email them the sequence of your heavy chain and light chain. From there, they will make the ADC, and off you go. And some of these molecules are now in the clinic in the hands of Exelixis Pharmaceuticals. Okay. What about cells? We can do chemistry on cells. We already know that. maybe we can add something to the world of cellular therapeutics. So CAR-Ts, everyone's really excited about this new form of immune therapy. But as you know, it's expensive, it's hard to scale, it's very bespoke. The typical protocol involves first removing blood from the cancer patient, taking their T cells ex vivo, doing some genetic engineering to introduce a chimeric antigen receptor, which targets an antigen on the tumor, so that after you grow up these cells into the millions and then reintroduce them into the patient, in the perfect world, those T cells home to the tumor and kill it and do the job for you. And it's remarkable when these therapeutics work. And for certain blood cancers, you can get a cure with a CAR-T therapy. But, like I said, it's expensive, it doesn't scale, and it's kind of patient by patient by patient. So most people don't have access to this technology, even patients with cancers that, could benefit. Well, one of my former students formed a company called Asapodia, on which I serve on the board of directors, that is using bioorthogonal chemistry to chemically engineer T cells to home to tumors, rather than genetically engineer. As a consequence, they can take off-the-shelf T cells, the so-called gamma delta T cells, that are generally well tolerated in allogeneic hosts. So they don't get rejected by people, even if they didn't come from that person, right? So what they do at Asapodia is they take these off-the-shelf T cells, they do some bioorthogonal chemistry to introduce a tumor-targeting antibody from the outside, and in early clinical data, these engineered T cells are actually very effective at homing to and killing blood cancers in patients. So hopefully this will make a CAR-T-like therapeutic cheaper and more available to a broader spectrum of patients. And then finally, I mentioned early on that we had this dream of taking chemistry out of the round bottom flask and onto the cell, but can we go all the way to the chemistry in the human body? And the answer is yes. And this is now happening in the hands of a company in the Bay Area called Shasky Pharmaceuticals. So they're using a chemistry that's called the tetrazine ligation. It's kind of a cousin of copper-free click chemistry in that it also uses a highly strained reagent, but it's not a cyclooctine. Instead, it's a trans-cyclooctene, or a TCO. And these trans-cyclooctenes will react with tetrazine reagents. It's a cycloaddition. And you can maneuver the chemistry. So at the same time these two things are clicking together, there's the release of this side chain. So you can use this chemistry to not only click, but release, which makes it nice for a prodrug application. So what Shasky has done is they have taken chemotherapeutic drugs like doxorubicin, oldie but goodie, and, well, goodie, not so much for the patient, but hopefully for the outcome. And they modify that drug with the TCO molecule in a place that inactivates the drug. And then they have this protocol where they'll first inject into the intratumoral environment for a patient a hydrogel polymer that has the tetrazine anchored to it. And that on its own is inert in the human body. It does nothing, it just sits there. But then later, when you infuse this doxorubicin prodrug, whenever that drug encounters the tumor, it can react with the polymer through that click to release and spit out free doxorubicin at very high dose, very locally, just in the tumor. So you can have this prodrug coursing through the body, but it's not toxic. It's only toxic as it's released in the tumor microenvironment. And they're in a clinical study right now to see if this can be used to treat soft tissue sarcoma. So, here in the year 2025, I think it's certainly clear to me that what started as the solution to a specific problem, which was to image cell surface sugars, kind of took off and found all kinds of interesting applications, some in my lab, but most outside in lots of other settings. And most excitingly now, in ways that will hopefully translate into treatments for human diseases. So there's a lot of applications out there. There's a lot of new chemistries that have been brought to bear. And I just love seeing how the community has really run with this kind of science. So we can't predict the future, but I think bioorthogonal and click chemistry will be part of it, at least I would hope. And hopefully, we have some tools that could be useful to you as well. And if you see something here that you didn't know about that you want to try, let me know, send me an email. We'll send you whatever reagents we have, of course. So with that, thank you very much. I can't thank all the people that contributed to this because there were dozens of them over many, many decades, but I'm indebted to my students and postdocs for doing so much beautiful work with me. And happy to answer questions, of course. Thank you.
Thank you. Folks here, of course, use the mics. I have a quick one to get us. Oh, okay, please. Yeah, good.
Hi.
Hi.
Super cool. The thing I'm thinking about the entire time I'm watching is that 8-membered ring thing.
Yeah.
Is that thing? Where does that come from? You have to make it, and
Yes.
Is it unstable and expensive? That was what I was sweating about.
Yes. In the early days, there really wasn't much literature on cyclooctines. They were a curiosity from the 1960s. There were a couple of papers. People had played with them back then. So we did develop some synthetic routes to make them, to decorate them, to tune their reactivities over many orders of magnitude. Nowadays, you can buy all of these reagents from commercial sources. There is a company called Click Chemistry Tools that was kind of a mom and pop shop out of a garage in Georgia that was selling them. Until about 10 years ago, they got acquired by Vector Labs, which now sells them as well. But you can buy them from Thermo Fisher. Bachem sells a bunch of them. There's at least a dozen reagent companies. So all of this stuff is now totally commercialized. You might have already used these reagents as reagent A, B, or C in some kit that you bought. Sometimes you don't even know what you're... You can make them stable. The ones that we use are very stable. We store them at room temp on the benchtop. The more reactive they are, the less stable they are in general. So you can really soup them up so that they're super fast in their reactivity, but then they don't have a very good shelf life. But the ones that we use are not quite as fast. They're a little slower, but fast enough, and they're fine, yeah. We don't treat them in any special way with kid gloves or anything. And by the way, afterwards, come up and take a look at my earrings, if you can't tell from there.
Good, Dr. Banerjee.
Carolyn, that was a beautiful talk. This is about sort of new bioorthogonal reactions. So there is so much sort of chemical reactivity data these days that people are doing sort of retrosynthesis sort of, using AI. So I'm curious, is there a way to parameterize all of this reactivity data and then use the known bioorthogonal reactions to then predict what could be new bioorthogonal reactions? Do you think that can be done? Or is this being done?
It's a good question. So lots of people nowadays, more hardcore chemistry labs, are interested in developing yet next generations of bioorthogonal chemistries. Where they could be most useful is if they are orthogonal to the current set. So you could be running these reactions in parallel with each other at the same time without any cross-reactivity. Most of the work I've seen has taken the same theme that we took, which is dial the clock back to the 1950s and 60s, where physical organic chemistry was supreme, and people were just interested in weird, exotic reactivities. And you can find some pearls from that old literature and bring it up to the modern day and use it this way. I think the field is shifting now more towards actually using modern computational methods, because we actually have the ability now to simulate transition states, right? And this is all about transition states. You have two reagents. You want them to combine with each other in a transition state that is lower in energy than any possible transition state they could achieve with a biological chemical. That's the selectivity. And, I know some people that are modeling, like, okay, if I take this reagent and these thousands of slightly perturbed reagents, which one might form a low-energy transition state? I think that is a really interesting and potentially exciting way to break open new innovation in this space.
Thank you.
Yeah.
Okay. Let me sneak in one question from online. I'm in the front row, but you don't have to look at me. Over here.
I can't find you.
There you go, front row. Hey.
Sorry.
It comes from online, though, so you don't have to stare at me when you answer. Azides are known to be toxic to humans. So how would you study azides in humans?
This was like my first grant application to NIGMS back in 1999. So azide anion, N3-, the salt, is toxic. It's used as a metabolic poison. We put it on our FPLC columns to prevent bacterial growth. But organic azides, where the N3 is attached to an organic molecule, not a negative charge, but just a bond to a carbon, those are harmless, totally fine. And for example, the drug AZT, That was one of the first generation AIDS drugs. That has an azide. People take grams of it, or people took grams of it per day. And GlaxoSmithKline did all the metabolic profiling. The azide comes out in the urine, just unmetabolized. So they're actually very safe, as long as they're organic azides and not N3-.
Okay, bioorganic, got it.
Yeah, N3- is a poison to the electron transport chain.
Okay.
It was a very excellent talk. Yeah. The reaction of a click reaction proceeds by 1,3-dipole addition reaction of Husky. Who introduced it?
Who invented it?
Oh, we invented 1,3-dipole addition reaction.
Okay.
He was my teacher also in Munich University. Anyway, but of course it was a click reaction with the copper sulfate, and that you can make it at room temperature, not in the flasks of a temperature. Yeah. This was, and so you have set up the beautiful, fantastic direction of the chemistry drag. But one for the antibody-drug conjugation, we have developed methods where we have used the enzymes to connect with the sugars. Based on the structure, we mutated so that it can be basically connected with the via sugars, glycan sugars. That is not, you did not include none of this.
Other people have, yeah. So we developed this technology for antibody-drug conjugates. You might ask, why didn't we put an azide onto the protein? Because methods for introducing azides, like an unnatural amino acid into the protein, those exist. Peter Schultz's lab pioneered that kind of technology. There's companies now that use that. Those companies are putting azides into antibodies to make ADCs using unnatural amino acid mutagenesis, but it's a pretty complex engineering problem. You have to engineer a lot of systems to get cells to make high quantities of antibodies with unnatural amino acids. And so, quite frankly, we thought this was an easier way to do it, where the unnatural amino acid is not actually unnatural. It's a repurposed, known post-translational modification that creates a different kind of chemical group. So honestly, we just thought that we could get to site-specific ADCs on scale faster this way. But other Sutro pharmaceuticals, like if I go back to that slide with logos on it, whoops, sorry. Yeah, this company, let's see, Sutro, they're making antibody-drug conjugates with azide copper-free click chemistry. They licensed the Berkeley patent to do that. Ambrx, same thing. They're making site-specific ADCs with copper-free click chemistry. Synaffix, same thing. So at least these three are, in fact, doing that. But at Catalent, we have a different chemistry.
Our mutant enzyme is being used with the companies. And mutant transferase, which can then take a keto.
Yes.
And then
yeah, in fact, Synaffix does that kind of stuff. So Synaffix does glyco modifications of the antibody with azidosialic acid and click onto that.
I'd like to squeeze in 3 more questions, if you don't mind. 1, and then 2, 3. We're at the hour now, so.
So maybe I missed this, but on one of the on this side, on one of the last slides, when you were talking about the tetrazine ligation, I was wondering, like, what about the tumor microenvironment causes the release of the, causes the binding and release?
Yeah, sorry I went through that so quickly in the interest of time, but since you asked, now I can take more time. The idea is this. So this is a hydrogel polymer. It's a modified form of hyaluronic acid, which is Juvederm. It's a safe polymer. It's used for cosmetic filler. And they chemically modify it to have this tetrazine. And then they inject it into the tumor, so it's a blob of polymer in the tumor, sitting there, doing nothing. Then when that polymer encounters this prodrug, the tetrazine and the transcyclooctene find each other, and boom, the reaction just happens spontaneously. And when that occurs, all of this unraveling happens, and you just liberate the free doxorubicin, which is now toxic, but concentrated in the tumor microenvironment.
Thank you.
Okay. So 2 more, I'm sorry, but we will have a reception. Okay, go ahead.
Hello. This is a really general question, but to that point about the space of biologically occurring chemical reactions, I was just wondering what is known about how quickly biological systems evolve to explore new, chemical space, and I guess kind of thinking about...
if we wait long enough, will reactions evolve in nature? Who knows? That's a great question. Well, one thing I've learned is it's unwise to say nature never does X. Because I think what we know about what nature does and doesn't do is like a tiny little scratch on the surface of reality. So who's to say what cool functional groups do exist in the human body that we don't know about yet? And I guess, all the more reason to study human biology with chemical tools, I guess. Yeah.
So this is also a very broad question, but with all of these like beautiful cyclic molecules and everything and these reactions, once they actually occur and react, what's happening clearance-wise? Like with the bioazides, that's renally cleared, but are there common clearance pathways for a lot of these, or is that something that hasn't been explored?
I wish I had more of substance to say in response to that question. The honest truth is we don't really know how the adducts that we make are getting metabolized. They do get cleared. They disappear. Presumably, they get broken down into some fragment that just gets cleared by the liver or maybe through the kidneys and excreted out in the urine. But I can't say that we've done a very thorough analysis of metabolites. So anything I say would be pure speculation.
Fair. Thank you.
Thanks for exposing that ignorance. Yeah.
Okay, I think we're going to wrap it up. I invite you all to attend a reception that's going to be across the hall in the NIH library. And please join me in thanking Dr. Bertozzi for an amazing lecture.
Thank you.